Hello everyone, welcome back to Study Room. In this video, we will be studying about heredity. So let us see what heredity is and how characters or traits are inherited from generation to generation. So heredity and what are inherited traits? Inherited traits are those characteristics that are passed from parents to their children. So there are certain characteristics that are passed from parents to their children, and those characteristics are known as inherited traits. Now, for example, we have earlobes that are either attached or unattached. We have dimples, eye color, hair color, muscle structure. So these are the characteristics, these are the general characteristics that we find which are inherited generation by generation. So here you can see in the picture also, it is given here, this earlobe is attached and this is unattached. You can see here that these are the variations of the earlobe that are found in the population, that are found in the human population, right? And here you can see that this earlobe is attached and this is unattached. And when, if a parent has a unattached earlobe, then the offspring is also likely to have an unattached earlobe. But if a parent has attached earlobe, then the offspring is also likely to have a attached earlobe, right? And if a parent has dimples, then the offspring is also likely to have dimples. In the same way, if the eye color is black, like the pupil color, the color of the pupil of the eye of the parent is black, then the offspring's pupil color will also likely to be black. If the hair color of the parent is black, then the offspring's hair color will also be likely to be black. And if the muscle structure, so if a parent is generally thin and very lean, so the offspring is also likely to be thin and lean. But if a parent is very healthy, and then the offspring will also be likely to be very healthy. So this is how traits or the characteristics are carried on generation by generation, and these are the traits that are known as inherited traits. Genetics is the study of heredity, the expression of traits and how they are passed from generation to generation. The science behind inheritance was understood in the mid-19th century, around 1865. Australian biologist called Gregor Mendel, he published the results of hybridization studies of pea plant findings, and he noted that there were certain factors that influenced the expression of traits, and those factors were later came to be known as genes. So Gregor Mendel or Gregor Johann Mendel, here it is written as a short form, G.J. Mendel, is often known as the father of genetics. Okay, so he was a person who actually introduced us to this whole mechanism of genetics, that there are certain characteristics that are inherited generation by generation, and this concept was introduced in the modern world by Gregor Johann Mendel. Okay, so Mendel was a person, what he did was, he used a number of contrasting characters of garden pea plant, the garden pea plant species, and he performed so many experiments with this plant. And why did he choose this garden pea plant? He chose this garden pea plant because it produces a large number of seeds. Okay, there's a lot of seeds that are produced in the garden pea plants. It has a very short life cycle. Okay, and it is self-pollinating, and various contrasting traits can be observed in the garden pea plant. So these were the advantages. And why are these traits advantageous? Because while performing experiments, if the plant has a short life cycle, then he can grow and regrow the plant again and again within a very short period of time, right? So this is a very good advantage. The second advantage is self-pollinating. So what happens is when the plants are cross-pollinated, then it depends generally on the chance event, that whether the seeds are getting pollinated or not, whether the seeds are getting they are getting produced or not, whether the plant is getting pollinated, whether the pollens are getting pollinated or not. So these are the things that are chances. So here the pea plant is self-pollinating, right? So and it is self-dependent of reproduction, right? So that is how pea plant is very advantageous, because it can reproduce by its own. Now, the other characteristics is that it has various contrasting traits. So, since it has many traits, he could perform so many different experiments using those different traits. Okay, and it produces a large number of seeds. So, there's a chance of seed viability. There's a concept of seed viability, that how many seeds are getting germinated at last. There are certain seeds which get lost due to the environmental adverse conditions and certain things. So if very less seeds are produced, then there's this chances that the seed might not survive or might not germinate. But here, since a large number of seeds are being produced, so he was rest assured that the seeds will actually germinate, and there will be a lot of plants that he can cultivate in his garden. So this were the benefits due to which he chose the garden pea plant, right? Now, Mendel's contribution to genetics is very huge. Okay, and here you can see the characters. The different characters that are present, the different traits, the varying traits that are present in a pea plant, and which was very wisely used by Mendel in his experiments. So the different characters are, first, the flower color, which is purple or white. These purple is a and in this, here we have the flower color, we have the seed shape, we have the seed color, we have the pod color, we have the pod shape, we have the plant height, and the flower position. So these are the contrasting traits that we have, and these traits were very wisely used by Mendel in his experiments, right? Now, about how he did those experiments. Okay, so Mendel's hybridization experiments demonstrate the difference between phenotype and genotype. Now, what is this phenotype and genotype that we are talking about? So, phenotypes are the observable traits that are expressed by an organism and are referred to as phenotype, right? So, the observable traits, like whether the plant is tall or the plant is dwarf. Okay, so this was a observable trait. Like when you see the plant, you can just get to know that this is a trait, this is a character of the plant. Okay, so if a plant is tall, you can just see and say that this plant is tall. Okay, if the flower colors of the plant is purple, then you can just see and say that this plant has purple flowers. And if the plant has white flowers, then you can just see through your eyes, and you can naked eyes, and you can say that this plant has white flowers. So these characters are generally the phenotype. And now, what are genotypes? An organism's underlying genetic makeup, consisting of both physically visible and non-expressed alleles, is called its genotype. So genotypes are basically the underlying genetic makeup that gives rise to these phenotypes, that gives rise to these observable traits. So there are certain genetic mechanisms in the body of the plant or of the organisms, and these genetic makeup actually gives rise to these observable traits in those organisms, and this genetic makeup is known as genotype. Now we'll be studying more about these genetic makeup and everything, and if we tell what is the genotype of this tall and dwarf, so genotype can be capital T, this can be tall, and this can be small t for the dwarf one, right? So this is how the genotype and the phenotype actually works. So here we'll be discussing about Mendel's monohybrid cross. What did Mendel do? And this was an experiment that Mendel did. He did was with monohybrid cross. So he took pea plants with different characteristics. A tall plant, as you can see here, a tall plant was there and a dwarf plant was there, right? And he produced progeny by crossing them, okay, and calculated the percentage of tall or short progeny. Now, parental plants and F1 tall plants were led to reproduce by self-pollination, okay, and the progeny of the parental plants were all tall. So you can see here that how this is a phenotype, right? This is a phenotype here, and this is the phenotype here. So, and this is the underlying genetic makeup, that is genotype here. The capital T is small and small t is dwarf. Okay, so here we are producing the gametes. Okay, so this capital D will come here, this capital D will come here. So we have two gametes, that is one is capital D and this one is capital T, and then we have this gametes here, that is a small t and small t. So here the half number of things are coming, right? Half gamete. So half is coming. So half 1. Okay, and 1 is here capital T and small t. Here also 1 is coming. Okay, so capital T, and here also capital T and small t. So when they combine together, they form this F1 individual, that is known as that is a F1 individual, and it has the phenotype of tall. Why? Because we have this one capital T and this is a small t. And why? And he saw that Mendel observed that the F1 individual was tall. The observable characteristic was that the plant was seen to be tall. Now, why was this seen to be tall? That we will be discussing in the the later part of the chapter. But now we need to see that here he observed that this plant was tall and the genotype of this plant was capital T small t, right? And how did this capital T small t came? So here he was making a very good concept he developed, that was known as how he could combine the gametes. And this concept of this square concept, right? How we can combine the gametes and form the individuals, this is known as Punnett Square. Okay, so this is known as Punnett Square, and this concept was invented by a scientist known as R.C. Punnett. Okay, so R.C. Punnett was a person who actually saw how the genotypes, how the gametes are actually combined, and these genotypes are formed. So here you can see capital T and small t, they combine to form capital T and small t. Here you can see capital T capital T and small t, and this also combines to form here and capital T small t, right? So this is here 4 kind of progenies are getting formed, and all the progenies are of the same type, of the same genotype, and were observed to be tall, right? So this is how he saw the F1 generation. Okay, so in the F1 generation he saw that all the progenies were tall and the genotype of the progenies was capital T and small t, right? So in the next generation, what Mendel did was he took as parents. Okay, in the next generation he did the second generation cross, and he took the first generation cross result. Okay, that was capital T small t, that was tall plant. Okay, this was a tall plant, and he took another the same kind of progeny plant. Okay, that was also tall. So he did this, and this process is known as selfing. Okay, because we are taking both the plants of the same kind of progenies, that is the resultant progeny of the first generation, right? F1 progeny, we are taking the F1 progeny and we are selfing two progenies. And then what he found was, from here we had gametes. Okay, we had two gametes, here also we had two gametes, right? And this in the F2 generation, these are the gametes. Okay, so the gametes will form capital T small t, capital T small t. And the F2 generation, okay, F2 generation we saw that these gametes were combined. So what happened? We will make the Punnett square here. Capital T small t, okay, capital T small t. Here will give male, female one. And how we can make the Punnett square here? We will just combine, very easy, we'll just combine the gametes. Capital T. Here we can see capital T, small t, capital T and small t. Here we can see small t and capital T, right? And here we can see capital T, small t and small t, right? So this is how the progenies are formed. So here we can see that there are 4 kind of progenies, but ultimately in the form of genotypes you can see here only 3. Okay, so the genotypes that were obtained was capital T, that was observed to be tall. The next genotype that we saw was capital T, small t, that was also observed to be tall. And the third genotype that we had was small t, and it was surprisingly observed to be dwarf. So this is what the result of the second generation cross, what Mendel found out. Okay, This was the experiment. This was the whole experiment that Mendel did. And in the second generation, the F2 generation progeny, we can see here that one tall plant. Here you can see here only one T, capital T, capital T, and here two capital T and small t, two capital T and small t. This two are formed here tall, and this is also one small t, small t. So this is how this was the thing that Mendel found out. This was what he observed. Now we will analyze the results of Mendel's cross. Okay. So in F1 progeny, we saw that all the plants were tall, and this meant that only one of the parental traits was seen. Only one of the parental traits was seen. That means that we had two parental traits, right? We had two parental traits. One was tall and the other was dwarf. But in the F1 progeny, we had this progeny, right? And this was observed to be tall, and therefore only one parental trait was seen. So this was the F1 generation. In the F2 generation, the progeny of the F1 tall plants are not all tall, but 1/4 of them are short, and this indicates that both the tallness and shortness traits were inherited in the F1 plants, but only the tallness trait was expressed. So if you go just back and you see that in F2 generation we had three traits, right? So and 1/4 of that trait, okay, 1/4, we had one capital T, capital T, we had two capital T, small t, and we had one capital T, capital T. And this was dwarf, and this all were tall. So this large chunk was tall, and this one chunk was dwarf. So here they are saying the same thing, that 1/4 was found to be short. And this indicates that both the tallness and shortness were inherited in the F1 plants. As you can see here, both tallness and shortness genes are inherited in the F1 plant, F1 progeny, F1 generation. But they are not shown in the F1 generation. What the observable characteristic that we are seeing in the F1 generation was tall. But in the F2 generation, you can see that both the characteristics are expressed. Okay. While discussing the monohybrid cross, I said that the first generation progeny was observed to be tall. But why was it tall? I said that the reason about why was it tall, I would discuss it later on. So here comes the reason that why the F1 progeny was observed to be tall, and why there were the expression of both the characters in the F2 progenies. Okay, so here we see that the dominant trait, okay, the dominant and recessive traits. Okay, so the traits that are dominant, the trait which appears in the F1 progeny is said to be dominant. So what was the F1 progeny in the monohybrid cross that we saw? The F1 progeny was capital T, small t, and the observed character was tall. So tall here is the dominant trait, and capital T is the dominant allele. On the contrary, the trait which remains hidden or which does not appear in the F1 progeny is the recessive trait. So what is the trait that is remaining hidden here? That is the small t. Okay, so small t is the recessive allele, and the character, the trait that is recessive here is dwarfness, right? So dwarfness is the recessive trait and tallness is the dominant trait. Now dominant trait appears in more number, as we saw in the F2 progeny that there were 3 tall and 1 dwarf. So we can just see that 3 tall, and that is why it appears in more number, and 1 dwarf, so it appears in less number. So the dominant trait was tallness in the monohybrid cross, and the recessive trait was dwarfness in the monohybrid cross. And here you can see that the dominant trait is the trait which is appearing in the F1 progeny, and the recessive trait is the trait that is hidden in the F1 progeny. So there is a very simple difference that we can see, and the example can be the example of monohybrid cross. Now Mendel's experiments and the results that he got, there were two copies of factor now called genes controlling traits are present in sexually reproducing organisms. Okay, so these are the factors, or there are certain genes which control the expression of the traits in the organisms, okay, in the sexually reproducing organisms. And these two may be identical or may be different depending on the parentage. An allele is one of two or more versions of a gene, and individual inherits two alleles from each gene, one from each parent. Okay, so allele is basically one of two or more versions of a gene. Okay, so now as we saw that this genotype we saw, right? We saw this genotype. There were certain genotypes that we saw, right? And these genotypes actually result due to the expression, due to the controlling of these factors, or the controlling of these genes. And here you can see that two chromosomes are there. One of them, on these chromosomes there are genes, okay, and these genes have, they have two copies, or we can say which are known as the factors, and one of these is known as allele. Okay, so this allele is basically one of the two or more versions of a gene. An individual inherits two alleles from each gene, one from each parent. So one allele is inherited from the father, and one allele is inherited from the mother, and hence the individual will basically have two alleles for each gene. Okay, so the gene has two characters, two alleles. A gene consists of two alleles, in an individual. Okay, in an individual a gene consists of two alleles. One of this is inherited from the father, and one of these is inherited from the mother, and thus combine to have two alleles in the offspring. Okay, now dominant or recessive alleles. As we discussed about dominant and recessive traits, we'll be discussing about dominant and recessive alleles too. So dominant alleles is able to express itself even in the presence of the recessive allele. I had already discussed about this allele thing in the dominant and recessive traits, but here again I'll say you, the first generation progeny of the monohybrid cross had capital T and small t, right? So the small t was the recessive allele and the capital T was the dominant allele. Why? Because this capital T is getting expressed, okay? It is expressing itself in the first generation progeny, while the recessive trait, that is the small t or the recessive allele, is remaining hidden. So the recessive allele is unable to express its effect in the presence of dominant allele. So when this capital T is already present, so there, it cannot show. It is always being tall. Always, when there is a dominant allele and a recessive allele, in the presence of the dominant allele, the recessive allele gets suppressed, and the character that is expressed will always be of the dominant allele, that is tall. Okay, and it does not require another similar allele to produce its effect in the phenotype. So the capital T and small t is tall. Here it is written small. No, it's tall. Okay, it's tall. The capital T and small t is tall, and here it is written for the recessive alleles that it produces its phenotype effect only in the presence of a similar allele. That is, when there are two small t, only then we will have the dwarf character, right? So we had seen in the second generation progenies that when there was small t and small t, that is, this is the recessive trait, this is the recessive alleles, when two recessive alleles are present, only then the recessive character, that is dwarf character, can be seen in the plant. So this is the role of alleles, and this is how, due to the alleles, due to the expression of these alleles, we have the dominant and the recessive traits. Now, Mendel's experiment of dihybrid cross. Earlier we studied about the monohybrid cross, in which we had only one character. We had considered only one character, that was tallness or dwarfness. That was the plant height. Okay, we considered plant height, right? We considered plant height in the monohybrid cross. Okay, but here in the dihybrid cross, we are considering two characters. The first character is round and yellow, okay, and the second here the recessive one is wrinkled and green. But here you can see there are two characters. One is the seed color and the seed shape. Okay, the seed shape, the dominant shape is round and the recessive shape is wrinkled. Now you know that what is a dominant and a recessive trait, and what is a dominant allele and the recessive allele, right? So here the dominant trait is round and the recessive trait is wrinkled. Here in seed shape and in seed color we have the dominant trait as yellow and the recessive trait as green. Okay, so when these two, they are crossed, and then there is this production of F1 progeny. So F1 generation, what will be the result of the F1 generation? We will find that the F1 generation will have capital R small r and capital Y small r. Why? Because from here we'll get R and from here we'll get Y. From here we'll get small r. From here we'll get small y, and we'll combine all the four gametes, and this is the F1 progeny. And what will be the result of the F1? Like, what will be the observable characteristic or the phenotype of the F1 progeny? The phenotype of the F1 progeny, as we know, that it will be the dominant phenotype, necessarily be the dominant phenotype. And the dominant phenotype here was round and yellow, right? So we'll have round and yellow seeds. So then again we'll see here, we'll cross this generation. We'll do the self crossing in the F2 generation, as we did in the monohybrid cross. We did the self pollination and we did the self cross, and from here we will see, we'll make this whole complex Punnett square, where we'll get the gametes from here, and we'll just put the gametes here in the, and then the self-crossing gametes here, and then we'll receive these progenies. Okay, so here when we see the result, we see that there are 9 round and yellow, there are 3 round and green, there are 3 wrinkled and yellow, and there are 1 wrinkled and green. So you need to just remember that what is the ratio of the genotypes and the phenotypes. Okay, so here you can see that the ratio here is coming 9:3:3:1. So this is the ratio of the dihybrid cross. Okay, this is the ratio of dihybrid cross, right? And how is this ratio coming? We are seeing here with the number, that number of the number of characteristics that we can observe here. So the round and yellow seeds are 9, the round and green seeds are 3, the wrinkled and yellow seeds are 3, and the wrinkled and green seeds are 1. So combining all these ratios, we'll find here 9:3:3:1. Okay. Now I'll be discussing about Mendel's laws of inheritance. So Mendel had basically 3 laws of inheritance. The first law was the law of dominance. Okay, so it states that for a pair of inherited traits, the dominant allele will be exclusively expressed in the offspring instead of the recessive alleles, unless both alleles are recessive. So this is just the concept of the dominant and recessive allele that we studied before, that when there is this dominant and recessive, both the alleles are present, then only the dominant allele will be expressed. So this is the law of dominance, that only the dominant allele will be expressed. But if there are both the alleles that are recessive, okay, if both the alleles are recessive, that is small t, only then we will have the recessive character. So, but if both the alleles are present, then we will necessarily have the tall character, which is the dominant character, and that is what is the law of dominance. You can just connect it with the example of the monohybrid cross to understand it better. Here the definition that is given, it explains the same, that the dominant allele will be exclusively expressed in the offspring instead of the recessive allele unless both alleles are recessive, right? The second law that we have is the law of equal segregation. The two alleles or the gene pairs of a hereditary trait separate during the formation of gametes and are then carried by different gametes. As we saw that there were these gene pair, right? Capital T, small t, small t. And when we formed the gametes, then we had these two gametes, and then we had these two gametes, right? So these are the gametes that are forming, okay? And these are forming equally, right? There are equal segregation that is going on here. The distribution is equal within the two gametes, right? The distribution is equal, and this character, this whole concept is known as the law of equal segregation, where the two alleles of a hereditary trait are separating from each other during the formation of the gametes and are then carried by different gametes, okay? Different gametes. So this is the law of equal segregation. The third law that we have is the law of independent assortment, and we will understand this law with reference to the Mendel's dihybrid cross. The first two laws we understood in reference to Mendel's monohybrid cross. We gave the example of Mendel's monohybrid cross, but in the third one, we'll understand it with Mendel's dihybrid cross. Now, alleles of different traits get assorted independently of one another during the formation of gametes. Alleles of different traits, okay? In the equal segregation, law of equal segregation, we had alleles of same trait. But here we have the alleles of different trait. That means, in the dihybrid cross, we saw that there was round, yellow. There was this wrinkle, and green. So this trait is different, this trait is different, this trait is different, this trait is different. Now how will this alleles be segregated? We'll have one gamete from here, we'll have one gamete from here, we'll have one gamete from here, we'll have one gamete from here. So all these gametes are of different traits. As you can see here, the gametes are of different traits. This is of round, this is of yellow, this is of wrinkle, this is of green. So these are getting assorted independently, and this concept of assortment of the gametes of different traits, the alleles of different traits into different gametes is known as, and assorting them independently. This concept is known as the law of independent assortment. Okay, and the genes are located on different chromosomes and will be inherited independently of each other. That is why, since the genes are located on different chromosomes and are inherited independently of each other, that is why there is this independent assortment of alleles of the genes, and these alleles form the gametes. Okay, the independent assortment and the independent gametes are formed, and this is basically due to this reason that the genes are located on different chromosomes and will be inherited independently of each other, which will give rise to the law of independent assortment. So these were the three laws of Mendel's, which are really important, and you will need to express and you will need to write them and explain them in your exams with proper and suitable examples. Okay, so this is how you can just explain it, and the concept is really easy if you can just understand it properly, and for once you can just write down and see whether you are understanding it properly or not. The next topic that we have is how are traits expressed. So we understood that different traits are expressed differently, and we have different dominant and recessive traits. But what is the genetic makeup, or what is the genetic underlying genetic reason that we have? The reasons that we have is discussed here. A segment of DNA provides information of one protein and is called a gene for that protein, right? So there are proteins, and the DNA produces these proteins, and the information is stored in a gene, okay? And these genes influence the traits, as we saw before also, that the traits are actually resulting due to the genes, okay? So each trait is the outcome of biochemical reactions. Each of this is controlled by a specific enzyme, okay? So these traits are basically the result of biochemical reactions which are controlled by enzymes. And each parent contributes one copy of the gene for a particular character, as we saw that each the alleles, one parent contributes one allele and the other parent contributes other allele, and then they both combines and gets into two alleles, which is known as a gene. Okay. In this video, you can see that each human has between 20,000 to 25,000 genes. This collection is called the genome, and which determines a person's traits by influencing characters or factors on a cellular level. Okay, and the genetic information is stored in every cell's nucleus. Structures called chromosome carry this information in the form of deoxyribonucleic acid, or DNA, as we know. So you know that the genetic information is stored in the nucleus of the cell, and basically in the chromatin material that is present in the nucleus, which actually consists of the chromosomes in the condensed form, right? So how are the traits expressed? Again, we know that the Mendel proposed that paired unit factors were transmitted from generation to generation by the dissociation and reassociation of factors during gametogenesis and fertilization. Mendel deduced that hereditary characters are inherited as discrete units, and these units were known as genes. Okay, and a gene is made up of short sections of DNA that are contained on a chromosome within the nucleus. Now, Mendel proposed that unit factors were transmitted from generation to generation by dissociation and dissociation of factors, as we know, and it happens during the gametogenesis and fertilization, that is basically during sexual reproduction. Okay? So this is happening, this whole variation and the transmittance of the inherited traits from one generation to generation is basically happening due to sexual reproduction. Now we'll be studying how this is happening. Okay, so the genetic inheritance begins at the time of conception, right? And every germ cell will take one chromosome from each pair. That is from mother and from father. And these may be of either maternal or paternal origin, as we know. And when two germ cells combine, they will restore to the normal number of chromosomes in the progeny, ensuring the stability of the DNA of the species. This basically happens, and this basically is referring to the process that we studied in sexual reproduction, which is meiosis, or the meiotic division that takes place in the cells. Now, suppose you inherited 23 chromosomes from your mother and 23 from your father, right? And that is how, combining the 23 from the mother and 23 from the father, you are getting 46 chromosomes in your body, in your genetic makeup, right? And such a mechanism of inheritance explains the results of the Mendelian experiments and is used by all the sexually reproducing organisms. Okay, here you can see that how meiosis is happening. So here we had full chromosomes, and then they are forming duplicate chromatids, and they're getting condensed further, and then they're getting divided. Okay, so then they're getting divided, and then they're producing here with the parent, one from one parent, each parent, and then it will combine. This will have half the DNA content. If this has full DNA content, this interphase stage has full DNA content, then this daughter cells will have half DNA content and will have chromatids, right? Now let us discuss the in-text questions. The first question that we have is, how do Mendel's experiments show that traits may be dominant or recessive? Mendel took pea plants with contrasting characters, that are tall and dwarf, and on cross-pollination he got all tall plants in first generation, but in the self-pollination of F1 tall plants, the plants of second generation consisted of tall and short plants in the ratio of 3:1, as we studied before, right? And on the basis of these experiments, on the basis of the monohybrid cross of Mendel, the characteristics that appeared in the first generation were called dominant, right? And the characteristics that did not appear were known as recessive. The dominant character that appeared in the first generation was tall, and that means that it was a dominant character. And the character that did not appear in the first generation, that is recessive, which is the dwarfness of the plant, and hence dwarfness can be termed as the recessive character, right? The next question that we have is, how do Mendel's experiments show that traits are inherited independently? Now Mendel took two pairs of alternate expression of two traits and carried out the dihybrid cross by crossing them, right? In Mendel's dihybrid cross, we had two traits, and the traits appeared in first generation were termed as dominant. When he used these F1 progeny to generate F2 progeny by self-pollination of different types of by self-pollination of the F1 progenies, then we found that there were different types of plants that were produced in the F2 progeny of the dihybrid cross. In some plants, both the traits were dominant, while in some both were recessive, and some plants exhibited mixed traits, right? And this indicates that the traits are inherited independently. Also, we can refer to Mendel's law of independent assortment, where he says that the characters or the traits that are inherited are assorted independently during the formation of the gametes. Now we'll be learning about the sex determination in humans, how sex are actually determined in humans. So the genes inherited from parents decide the sex of the offspring. Now if the female, if the offspring has X and X chromosome, then it will be a female, okay? And if the offspring has X and Y chromosome, then it will be a male. So this is the basic difference between the male and female, and that is how the sex is determined in humans. All children will inherit an X chromosome from the mother, and the sex of the children will be determined by what they inherit from their father. Okay, since their father has X and Y chromosome and their mother has XX chromosome, right? So the mother will give X chromosome and the father will either give X chromosome or Y chromosome. So it will depend on the father. Mother is essentially giving X, right? But it will depend on what chromosome is coming from the father. Okay, what chromosome is coming from the father, and that will actually determine the sex of the child. You can see here that the male is giving X and Y, female is giving X. Essentially, if the X and Y are basically determining the sex of the offspring. So if it is XY, then we'll have the male, and if it is XX, then we'll have the female. The third question that we have is, a man with blood group A marries a woman with blood group O, and their daughter has blood group O. Is this for information enough to tell which of the traits, blood group A or O, is dominant, Y or Y? We can say that the information is not enough because each individual is carrying two alleles. The recessive trait can occur only when two alleles are similar, right? And in blood group A is dominant and O is recessive, then daughter can have both. Then the daughter can have blood group O only when both recessive alleles occur together in mother, and father has one allele of O and other of A. So here we can say that the information that is given is insufficient. The next question that we have is, how is the sex of the child determined in human beings? We just discussed just now we discussed that the X chromosome is coming, half of the X, like half of the male, the gametes of the offspring, the X chromosome is coming from the female or the mother, okay? And the sex of the child actually depends on the kind of chromosome that is inherited from the father. So the mother has XX and the father has XY. So whether the father will give X or Y, that will actually determine the offspring's nature. Okay, so here either it will be X or will be XY. So if this is XX, then it will be a female, and if it is XY, then it is a male, right? So this is how the sex is determined in human beings. So that's all about the heredity. In detail we discussed about Mendelian genetics and Mendel's experiments, the monohybrid and dihybrid cross, and the laws of Mendel's, and everything about sex determination in human beings, and how the genetic makeup is actually giving rise to so many traits. So these all things were discussed in details, and I hope that you followed the whole lecture. Thank you so much.